Picea. I'm a postdoc working with Zhongli Wang and Greg O'Neill. Thank you very much for having me here today. And it's very happy to share with you one of my postdoc research. It's about the local adaptation of trees to forest pests. I'm going to spend a couple of minutes to introduce the general background of this research, and we will make a very brief explanation for what methods we have used and what is the most important findings of this research. And I will wrap it up with the potential implementation of this project. So first thing first, what is local adaptation, and what is, first of all, what is adaptation? I know everyone here is an expert, but it's never a bad thing for us to review some basic concept from time to time. So adaptation, that is evolutionary process through which the organism change their biological structure, their behaviors, their physiological process, et cetera, so that they will have a higher likelihood to survive and produce the next generations. And what is local adaptation? That is a more specific type of adaptation. There are many populations within the same species, and if we pick up one populations and plant them in a wide range of the climatic niche, for example, they can survive, but it doesn't mean they will perform in their best situations everywhere. Just like us, we feel most comfortable at home. And why do we care so much about them? Because when we are planting the trees, we need to match the optimal seeds with their most adapted environments. We have, for example, we have a seed lot. So what is the optimal cut block will be? Or we have a cut block, and what should be the optimal the seed lot that we should use? Especially when we have the climate change in our minds. And when the temperature increases, it's possible that they are not matched with their climatic niche anymore. Some of them may be still growing, but they will not be happy there. And some of them will probably just die. That is why we need the local, to study the local adaptation. There are many researches already done, but primarily based on the productivity of the forest. Some of them based on the phenotypic data, some of them based on genomic data. And among all of these research methods in this project, we are focused on the transfer functions. There are two types of transfer functions we used for this research. The first one is individual transfer function. This is the most simplistic version for the transfer function, and that will be the basics for other type of transfer functions. So for example, the individual transfer function, this example is based on the site called Dog Creek with the MCMT test mean coldest month temperature around -10 centigrade. We have a site here, and we plant many different populations. Each dot here is a population. And we can see there is a transfer curve, and the most important thing, there is a single peak here. And the x-axis is the provenance temperature for each populations. So this is the local populations that we are going to use for this specific site. And of course, one site will not be enough. We need to use a lot of sites, a lot of populations. So another type of transfer function will be used. That is called the pool transfer functions. So in these functions, all of these sites and their populations will be put together. And to account for the confounding effect of the variation for site potential productivity, all the fitness index for in this example, that is the tree height, will be standardized. That will be calculated as the proportional tree height compared to the mean height of the local populations. And in that case, we have this transfer curve. And at zero transfer distance, this standardized height, here is called height p-value, will be 1. And the rest of them, they are not local populations. So in many cases, it's like this. This is for tree height, so local population perform the best. The study that I just demonstrated is based, and many other relevant studies are based on the forest productivity. And why do we care about local adaptation for forest fire resistance? Many reasons, but the most important thing is we need to guide the seed selection for the reforestations. Tree height has been considered as the integration for many factors, including the forest health, but there are some limitations might be ignored and may or may not have some severe consequences in the future, because our seed selection in many cases is based on the provenance trials, and in boreal forests, their age is usually way younger compared to the rotation age, and it's possible that when they are young, they may not have been experienced through some severe infection events. So the population we have selected may be pest resistant, that site infected, or may not, because we did not deliberately inoculate the pest there. So we need to understand what is really going on there. So the objectives for this project is to assess if there is local adaptation for the forest pest resistance, and is that a universal pattern for different species, different infection traits. And based on that, we can calculate the safe seed transfer distance. We have to make sure they are not be transferred to Dawei. And at the same time, we also assess the cross-resistance of the trees to multiple forest pests, and if they have common climatic drivers for them. To accomplish these objectives, we have collected a large data set that includes 25 trial series for 6 major tree species, and that includes the interior spruce, that's primarily white spruce and Engelmann spruce, they're hybrid, lodgepole pine, and western red cedar and western white pine, Douglas fir, and Sitka spruce. Most of them are from provenance trial. Some of them also include the data from the provenance trials, and data is from the BC and Alberta government, and some of them provided by the forestry company. Some of them are from the literature. So there are 14 combinations of host tree and pest in total, especially for lodgepole pine. This is based primarily from Illingworth's trial. That's a very famous trial with lots of good paper published based on them. And the pest includes fungi disease, includes the insect attack, such as the mountain pine beetle. Sorry, what's going on? Such as the mountain pine beetle or needle blight, something. Yeah, so as I mentioned, there are 2 types of transfer function used. The first is individual transfer function, and then the put transfer function. I'm just go through this quickly. So basically what we did is, first of all, calculate the population mean infection rate, and we fit individual transfer function to each of those test sites. Some of those sites may have very low infection rate. That means the difference between the population's pest resistance cannot be told. So some of those sites removed. So the threshold that we use is 20% to 80% of the infection rate. That is the infection threshold that we use. And the tested sites with the infection rates within this threshold is retained for the following analysis. And we fit individual transfer function to each of those sites. This is the quadratic curve. And we should have a trough here because this is infection rates. And there are many sites, and it's not big enough for all of them, but this is what we did in the second step. And we standardized the infection rates by dividing them by the mean infection rate of the local populations. And we put them together, and we will have the transfer curve for the put transfer functions. So the y-axis, that's the standardized population means. And at the 0 transfer distance, and at 0 transfer distance, it is 1. So, and in the next step, we set up acceptable infection rates in this study, we used 25% of the infection rate. That is extra 25% infection compared to the local population that is acceptable. And we set up the, this is the safety transfer distance. So some of them are missing, sorry. There should be a confidence interval here. I don't know why in full screen it's missed. But anyway, within this safe transfer distance is the blue color, and outside is the red color. And main summer precipitation is selected for the Western gall rust infection. And we can see that with the increase of the precipitation, the infection rate is going to increase in the fast speed. But when we move them to a drier site, they are also going to increase, but in a relatively slow pace. And this makes sense because the fungi, they usually prefer the hot and wetter places. And we repeat the same method to all of them, and none of these have, oh, there is, but okay, the color is not good enough. There is confidence interval here. So it's based on the upper boundary of the confidence interval of 95%. So, so basically for all of them, we have found a significant, the poo transfer functions. It's, we tried quadratic, Cauchy, and Weibull function, all of them, and we select the functions that with the maximum R square, and we can, in every single one of them, we can basically find the trough or a peak, depends on what traits we are taking a look at. For example, for the single peak, that's the, here is the life current ratio. That's the alternative indicator for the Dawei needle blight. So yeah, so the pattern is significant. The local adaptation is there. And at the same time, we also assess the cross-resistance using the canonical correlation analysis. So the entire model here accounts for about 44% of the variation. First and second axis, about 40% of the variation. And what we can find from this one is, so the blue arrow, that's the climate variable. For example, the Western gall rust is primarily driven by the continentality. And the mean coldest month temperature is closely related to the needle retention and the Dawei needle blight. They are alternative indicators to each other, so they are on the opposite side. And the mountain pine beetle attack was measured after the needle retention measure. They usually attack the trees, which is taller, with more juicy leaves or trunks. So what we can find from this figure is that there is cross-resistance. The selection of one of them definitely affects the selection of another one, and they have common climate driver behind this. So the major conclusion is, first of all, we found local adaptation for the trees to pest resistance, and that they are closely related to climate variation. Oh, I don't know what's going on. And there is cross-resistance. And we also calculate safety transfer distance, and some of them are quite narrow, means we have to be careful of their transfer distance. And what we will do in the future is to incorporate both of them in calculating the safety transfer distance. So yeah, the safety transfer is not just based on productivity, but also consider the pest resistance. So, and finally, I'd like to say thank you to all the people, include the authors and Ministry of Forestry in BC, Alberta, and all the forest companies that provide us the huge data set. That yeah, that's all for my presentation. Thank you very much. Well, for pathogens, I'm afraid we are not able to identify their genetic structures, because that's not something that we deliberately inoculate. So we basically focus more on the local versus native versus non-native trees instead of the pathogens.
Yeah, I guess what I'm thinking is that with a native pathogen or pest,
Oh, I see. I think they are.
There was white pine blister at the very least.
Yeah.
Anyway, you may not be able to answer that right now, but it is an interesting, as I'm reading through the draft and whatever too, is one of the questions, and we can talk more. Yeah.
Okay, thank you very much.
All right, thank you very much.
Okay, thank you.
My name is Colleen. I was originally, there we go, a PhD student in Janice Cook's lab, which I'm going to talk to you a little bit about my PhD research. I'm currently now the project manager for the Tree of 4 project, which is kind of the next generation of this research, and I'll plug that again at the end. So I'm going to be talking a little bit about the role of ophiostomatoid fungal pathogens in overcoming lodgepole pine defenses during mountain pine beetle attack. There we go. So you've heard about mountain pine beetle a little bit already today, but mountain pine beetle is a bark beetle pest native to Western North America, and it is quite clever in that in order to attack healthy, large trees, it will use aggregation pheromone to elicit and to draw in other beetles to coordinate a mass attack to overwhelm host defenses and facilitate successful colonization. And so mountain pine beetle carries several fungal associates that include Grossmannia clavigera, which is commonly considered to be the most pathogenic of these fungal associates. And then one of the primary hosts in host trees in BC and Alberta is lodgepole pine. And so when mountain pine beetle and its fungal associates attack these trees, we see several defense responses, an arsenal of defense responses that are mounted by the host tree that include resin production, lesion development, biosynthesis of secondary metabolites within these lesions. And we see this in response to Grossmannia clavigera inoculation as well. As Janice mentioned, we use that as a proxy for mountain pine beetle attack. So we see a very similar response to mountain pine beetle attack with its associated fungi, as well in response to fungal inoculation. And so partly because these, the mountain pine beetle and its fungal symbionts are so closely linked and seemingly dependent on each other, and also because these defense responses are very similar, this brings us to a really commonly held belief slash opinion slash published statement that the fungal associates that are carried by mountain pine beetle that are introduced at the time of mass attack are contributing to overwhelming host defenses during that mass attack period. And I forgot to mention, so mass attack is very quick. It only takes about a week. And then longer term, there are other things. But during this initial mass attack, which really determines beetle success, it's commonly believed that the fungus was contributing to the host, overwhelm of the host defenses and successful beetle attack. And so this kind of gives us a nice hypothesis that we can test of, okay, we haven't actually looked at, is the fungus contributing? And if it is, what role is it playing? And so what we can do is look at, okay, well, what are the pine responses to the different antagonists, the mountain pine beetle or the fungus, and how are they the same or potentially different? And so we can look at how trees and plants respond to different antagonists and activate their defense responses using hormones. And so a lot of this work has been done in other plant species, especially model organisms like Arabidopsis. Not as much work done in conifers, which makes this a little bit more tricky, but we can use this as a basis of what we expect to see. And so, there we go. So for chewing insects like mountain pine beetle, this is expected to activate jasmonic acid production, which would then trigger jasmonic acid responsive genes that trigger jasmonic acid specific defense responses. But for things like necrotrophic fungal pathogens like Grossmannia clavigera, this is actually expected to activate both jasmonic acid pathways as well as ethylene response pathways. And so we can kind of look and see in our host defense responses, which one do we see when we've got the combination of these two? And so this is a bit tricky because this has not actually been well studied in conifers. And so we wanted to look at, well, what's going on with ethylene production, especially because this is a very important signature that would define this response. And so if we're looking at an attack, a tree that's attacked by mountain pine beetle, we can go ahead and test the classic paradigm. We've got introduction of the fungal associates by the mountain pine beetle as it's chewing. So these fungal associates are carried in structures in the mountain pine beetle's essentially mouth, behind their big teeth. These are the terms that I was given by the entomologist on the team. They carry the fungal spores here, and they're introduced to the tree at the time of mass attack. And so if we were to look at the responses of the tree during mass attack, we can expect these two options. So in response to a chewing insect like mountain pine beetle, if there was just the response to mountain pine beetle, we would expect just JA production. But if we see response to just the netrotrophic pathogen, Grossmannia clavigeria, for example, we would expect to see both JA and ethylene. So then we can conclude, when we're looking at the response to mountain pine beetle mass attack, if Grossmannia clavigeria is contributing during that mass attack and triggering host defense responses, then we would expect to see production of JA and ethylene. But if Grossmannia clavigeria is not contributing during mass attack, then we would only expect to see signatures of JA production. And so, okay, well, so testing this is actually a lot more than just going out and doing it. We had to set up a pretty complex field experiment, and we had trees that we baited for mountain pine beetle attack and carry the natural fungal associates that they were carrying. And then we also had trees that we inoculated with just Grossmannia clavigeria so that we can compare this. So these are carrying potentially, they are carrying fungal associates. We know that from other studies in these areas. And then we had to have, the proper controls, looking at wound response, and then just unwounded trees. And so this was really difficult to do. We actually had to run these separately so that we could ensure that these trees did not also get attacked. And so one of the ways that we did that as well, and also protect all our controls from attack, is that we had to wrap all of our trees, all of the trees that were not being attacked by mountain pine beetle, with these aluminum mesh cages that went up to 3 meters. And this was a lot of work and a lot of aluminum mesh. And we had to monitor them regularly to make sure that they weren't being attacked. And we actually, despite this, we still had a couple of trees that we ended up losing, but we were fortunate that we had enough replicates that this didn't have a huge, huge impact. And so from this experiment, which was really successful, may never work ever again, which is why we still continue to regale people with this story. We were able to collect phloem and xylem from the tree. So essentially phloem is the bark, and then the xylem is the inner wood, so that we could look at, okay, what is the response of the trees in these situations. And so this is showing the hormonal responses, and I'll walk you through it. So essentially, we've got phloem responses and xylem responses, and then this is in response to Grossmannia clavigeria inoculation only, and then this is in response to mountain pine beetle attack carrying its natural fungal symbionts. And so we've got 2 different time points just because we wanted to make sure that we captured mass attack within that first week, whereas with Grossmannia clavigeria, we wanted to get the same time point that we could compare, but we also wanted to look at longer-term responses. And so this is comparing to a mock control, and I've just used arrows to indicate where we see a significant change, so either significant increase or significant decrease. And the main thing that we can take away from this is that we are seeing, I should also mention, so this is looking at jasmonic acid, and this is looking at ACC, which is the precursor for ethylene. So ethylene is a gas, it's very difficult to measure. This allowed us to measure these levels in the host tissue. So we can use this as a proxy. And so the main takeaway is that, especially in phloem, that we see in response to Grossmannia clavigeria, like we expected, increases of ethylene and jasmonic acid, and we're only seeing an increase of jasmonic acid in these mountain pine beetle attack trees. So that's really interesting. And we see somewhat similar responses in xylem, but it's a little bit more complicated. It's also a little bit further into the tree that may not be as impacted as quickly in the time frame of mass attack. And so if we go back to that classic paradigm, we saw for mountain pine beetle attack trees, even carrying their fungal symbionts, we only saw a response of JA production increased. And so from this, we can actually conclude that G. clavifera does not appear to be contributing to mass attack within the mass attack time frame. It does not appear to be contributing to overwhelming host defenses based on these hormone signatures. And so, okay, but why are we seeing similar defense responses to the fungus as well as to mountain pine beetle attack? Like what's triggering those lesions? And a main component of those lesions is phenolic compounds. So I wanted to look more at what was going on with those defense responses. I looked at a wide array of phenolic compounds, which there's lots of phenolic compounds that have lots of different functions. And so we focused in on looking at kind of the composition of the tissues of these trees to see if we could see differences in just the overall composition of phenolics rather than looking at a specific one. And so what this is showing is each point, we've used a multivariate analysis to kind of plot each individual sample and then look for patterns and relationships related to the treatments. And so we've got phloem and xylem. And so interestingly, what we saw for phloem and xylem is that for the most part, all of our treatments kind of grouped really close together, and those samples didn't seem to be super different in terms of phenolic composition. But what was really interesting is that in xylem, which is where we actually see the majority of lesion development, we actually see a much more different phenolic profile in the G. clavifera inoculated xylem relative to mountain pine beetle attack or control. And so this is showing that these are responding much more strongly, and phenolic biosynthesis is much more different in xylem of fungal inoculated trees. There we go. And so then, okay, so I wanted to look at, well, what's going on in terms of phenolic biosynthesis? We know that the profiles are different. Can we potentially pinpoint maybe some genes or compounds of interest? And so this is a very simplified version of the phenolic biosynthesis pathway, just showing 3 of the genes. And I've grouped I've shown just the groups of compounds just to give you an idea. But this is, if you were to look up this biosynthesis pathway, it'd be far more complex. And so I focused in on these 3 genes and looked at their gene expression in response to these different treatments. And so this is a bit more to look at, apologies. But essentially, yeah, these are the 3 different genes that are all really important in the phenolic biosynthesis pathway. Again, looking at phloem and xylem. And we see, a increased gene expression. So we've got 2 different controls here, and then this is the one that we want to look at. So we've got an increased expression of all 3 of these genes in response to Grossmannia clavifera. And we do see an increase in one of the genes in response to mountain pine beetle. A little less going on in xylem, which is really interesting. But we see differences between the severity, or not severity, but intensity of this response to Grossmannia clavifera relative to mountain pine beetle. It's much stronger in response to Grossmannia clavifera, even at the same time point. And so, in summary, thank you. So, initially, our question and the classic paradigm that we're testing is whether or not the fungal symbionts that are vectored by mountain pine beetle contribute to overwhelming host defenses during mass attack. And so we provided the first evidence to date that conifers synthesize ethylene. Previously, this hadn't been studied. Mostly the work had looked at gene expression. And so we are able to say that conifers synthesize ethylene. And because we were able to do this, we were also able to look at, okay, how does that signature potentially differ between these 2 responses? And so this ethylene signature allowed us to say that mountain pine beetle fungus associates do not contribute to overwhelming host defenses during mass attack, during that initial mass attack period of about a week. Definitely there are differences if we were to look over long-term responses, but during mass attack, they are not contributing. And we have some evidence that the tree is perceiving mountain pine beetle attack and G. fissure infection differently and activating different defense profiles. Yeah. And so with that, I think I'm actually, good on time. I do want to do a quick thank you. This was a massive amount of work, and I'm very grateful to my field crew, my field crew, as well as our collaborators and all of the members of the Cook Lab that, past and present, our funders. And then I also do want to do a really quick plug as the project manager for TRIA 4. So we are hiring a couple of positions. We've got a couple of others, but I thought these would be really pertinent to some of the members here. So we are looking for a bioinformatician postdoctoral fellow that might be of interest to some of you or your students or colleagues. And then we're also looking for another project manager that would work with me at Carleton University. And so these are newer, so I don't have the posting yet for this one. But if you want to talk to me, I would be happy to send that information along as we get it. And yeah, that's it.
Thanks, Colleen. We have time for one really quick question as the next person sets up.
Yeah.
Barley?
Excellent talk. I really liked it. I was just wondering, so you did the fungal pathogen on its own, and you did the bark beetle and the fungal pathogen. Is there any way that you could have looked at just the response if only the bark beetle was there? Could you grow the insect in the lab, for example, and culture it without the pathogen? I don't know if that's possible.
So people have tried to do that, and those beetles are usually just not very healthy or they die. So it does seem to be dependent in some way on the fungus during its life cycle.
Okay.
Yeah, so yeah, that would be difficult. And like, even if we could manage it, I don't know if I would trust them to be like as healthy.
Yeah.
Cool if we could do that later. Yeah, yeah.
Thank you.
Yeah.
All right, while we do a quick switcheroo, we have Barley Rose Collier Harris from the University of Oxford. That's all right.
Hi, thank you. No? Okay, I think I've got the hang of it. I'm Barley Rose. I'm a 3rd/4th year PhD student at the University of Oxford, which in the British system means I'm in my final year, so I should submit next year. And I'm going to talk to you about one experiment that I've done over the course of my PhD. I'm in John Mackay's lab. Some of you may know him. He was based in Canada before he moved over to Oxford. And I'm really excited to be here because I work on Sitka, which we grow a lot in the UK, but obviously isn't native to the UK. So for me, this is my first time seeing Sitka in situ, and it's great to talk to lots of you about other conifers that are growing over here in Western Canada. My talk today is looking at the transcriptional responses to multiple attacks by Hylobius abietis in Sitka spruce. So I'll give you some background. I won't give you that much background on Sitka. It's grown a lot here, but I'll give you a little bit of context on how we grow it in the UK. But first, I'm going to talk a bit about Hylobius, or as I will refer to it throughout the rest of the talk, the large pine weevil, which is this insect here. It's about 1 centimeter, and it eats bark. And the reason that it's a huge pest in the UK, but also over large swathes of Europe and into parts of Asia, is that when you plant seedlings out, it eats the bark ferociously. It will then girdle the stems of the trees and kill them. So in the UK, if we don't, at the moment, the main treatment is pesticides, and if we don't treat with pesticides, at least 60% seedling death, but frequently up to 100% seedling death. And part of that is the fault of the way we do forestry. Clear-cut forestry, which is the system that we use in the UK, is altering the population dynamics. The insects themselves are attracted to the smells released when the seedlings are cut down and the secondary metabolites. And so by the way we do this, we have a whole forest, we cut them all down. There are then loads of stumps so that insects can be attracted. They can fly up to about 12 miles So they can fly a really long way to come into the site. Typically, you'd have one tree, not a huge amount of secondary metabolites released, and so that not necessarily that many insects would appear, but as it is, we cut down huge areas, loads of the insects fly in, and they then breed. They live in the stumps, eating the root tissue, and they develop for up to 3 years. And there's this huge population for up to 3 years of development, and then the insects themselves can live for another 3 years. So you've got a really dense insect population for potentially a really long time. So leaving sites clear obviously isn't financially viable, but it definitely isn't financially viable because you'd need to leave them as clear, unused sites for nearly 6 years. So instead, we use pesticides. The pesticide that's used the most at the moment has just been informed that it's going to be banned from sale by July this year and banned from use in the UK by July 24. So suddenly, we need a new system. And there are lots of things being looked into. We're looking at nematodes as biocontrols. People are looking at different systems of growth. So if you scarify the land, if you mound, if you do different things during planting. But this is another way we're looking at, and I'm looking at potentially developing a resistance breeding program. It's a long way off. I'm the first step in the process. Sitka in the UK, third biggest crop by land area. 50% of the conifers that are planted in the UK are Sitka, and it contributes about £1 billion to the UK economy each year. Big, important crop. So, as I said, overall in my PhD, I'm looking to see if there could be heritable underlying resistance to Hylobius in Sitka, with the aim that then potentially there will be a resistance breeding program. I've done several experiments. Oh, that's come up really faint. I'm sorry. On my slides, you can see it, but I think because of the lighting, you can't hear. I've done some damage response experiments using UK material and also using material that I've kindly been provided by people here to look at seeds from all over the native range, because UK Sitka all originally came from Hylobius. Similar climatically to where it's being planted in the UK, which is why it was selected. But as you may know, the white pine weevil Sitka there is very susceptible to. And so given that that's also a bark-eating insect, there's potential that we're not getting the full genetic diversity that we could have, which is why I've done those studies. But this was using UK Sitka, this study I'm doing here. And I'm testing the hypothesis that Sitka might respond chemically and physiologically to Hylobius, and I've done that using an RNA-seq study. My study was in a greenhouse. I had tubes around my seedlings, and you put, oh, wrong way. You put a weevil in a tube, and I left it for 3 days, and I measured the area of damage. The way I measured the area of damage was pretty rudimentary. I had a piece of paper with different shapes of known area. This is in millimeters squared. I held these shapes up to the areas of damage and said, yeah, that looks about right, and recorded that number. So for the experiment, overall, I had 4 blocks, 3 treatments, 3 time points, and 6 families, so 216 saplings. I didn't use all of these for the RNA-seq, but I did use all of these to get this phenotypic data. My sampling setup, my 3 treatments were as such. My control received no insect attack. 1 weevil attack. Here's my weevil. It gets placed on the tree in the tube for 3 days. I then sampled over a time period. So I did one sampling directly after that, 1 sampling 3 days after the weevil had been removed, and 1 sampling point a month after the weevil had been removed. The third treatment was to look at priming. Priming is the idea, much like vaccines, that if you pre-treat something, either with a chemical or here with a previous attack, that potentially the subsequent attack will get a faster and more sustained response. So how I did this, rather than just having a weevil once, I first put a weevil on the tree for 3 days. I then removed it and let the tree grow for a month. I then again put a weevil on the tree for another 3 days and took at the same time points. So directly after this second period of attack, 3 days later, and a month later. So that's the setup. When I analyzed the RNA-seq data, I did it in 2 separate groupings. So I compared the control and 1 weevil attack, and I said that that would tell me about the plant's physiological response to weevil attack. And then I compared between 2 weevil attacks and 1 weevil attack, and I said that any differences there would be due to this priming effect. So my phenotypic response. This is how much damage the weevils did in that period. Nothing. There was no difference. So I've primed the trees with this previous attack. Did I see differences in how the weevils attacked the tree? No. Pretty even. If there was 1 exposure or 2 exposures, the area consumed in that second 3-day period was about the same. That's not necessarily unexpected. I was hoping to see an effect, obviously, but not unexpected because, as I say, it's a non-native tree with a native pest. They've not co-evolved, so it's possible that they're not able to respond to each other. Also, Hylobius is a generalist. It prefers conifers, but it will eat broadleaf bark if nothing else is available. So even if this tree has a quite strong defense mechanism, if it's such a generalist, it can eat so many different species, it might not respond to these different defense strategies. But then we look at the RNA-seq data. RNA-seq is a technique in which you look at which genes are turned on and which genes are turned off in a plant at a particular time, and you do that by looking at how many transcripts of the gene you find when you extract the RNA. So if there are many transcripts of the gene, it is switched on, it is currently being actively transcribed. If you don't see any, it isn't. So this is a Picea, a principal component analysis. The principal components don't mean anything. They just are a way to help spread out the data to see if you can see any clustering. And hopefully you, like I, can see clustering. It's really nice, and I got very excited. This is between 1 weevil attack and no weevil attack, so we're saying, is there an effect of weevil attack on the plant's response? Yes. 1 weevil attack, we've got a cluster of triangles over here. 0 weevil attacks, we've got a cluster of circles over here. What's interesting is you do see that there are 2 triangles in the circles area, but they are both from time point 3. So that time point a month later. So potentially the plants did respond, but by this 3rd time point, you're no longer seeing that response. And you can see as well that the strongest response that we see with these triangles is in time point 1. So the yellow triangles are the furthest away. So it's quite nice. I was quite excited. We're seeing some differential expression. It's also my first statistically significant results of my PhD, so I was very excited. And then I've got some tables. I'll just chat you through them a little bit. So when I see how many differentially expressed genes we have that are statistically significant with an adjusted p-value of under 0.05, for treatment, overall, we've got 4,000 genes that are differentially expressed. Of those, 1,600 are upregulated and 2,400 are downregulated. We also see a response of time. That makes sense. Over a month. It's a month during their growing period. You're expecting to see some differential expression over that time. We've also got a very small number of genes that are differentially expressed based on the interaction between time and treatment. I haven't dug that deep into that. It's interesting that they're there, but given the large numbers we're comparing this to, 79 doesn't seem like a huge number of genes at this stage. I then broke this down further. So rather than just looking at between weevils and no weevils, I looked at between weevils and no weevils at each individual time point. And I found this really interesting. You can see that at time point 1, immediately after the attack, that's when we've got the most differentially expressed genes. And interestingly, you've got a large number of them that are upregulated compared to downregulated. Whereas by the time we get to time point 3, we've got less differentially expressed genes. But of those differentially expressed genes, the ratio is the other way around. You've got more downregulated genes and fewer upregulated genes. So this could suggest that you've got a response, lots of genes are turned on when the weevil is attacking, and then by the 3rd time point, you're seeing downregulation at that point because then you've accumulated whatever you've accumulated, and so you're now having to turn off those genes after this attack. The attack's no longer going on. They're being actively downregulated. I then did a overrepresentation analysis using a system called David, which is where you basically take the list of genes that are differentially expressed, compare it to the whole transcriptome, and say, are there more genes in this list that have certain characteristics than you would expect? Or is this a random subset? Have we just said, normally, 15% of your genes are based on defense responses, and within this list, 15% are also based on defense responses? The answer is no. We have some genes that are overrepresented, and some genes that are overrepresented that are exciting. Genes that are involved in defense responses. Interestingly, here it says defense to bacteria. The way this works is that it looks at whole lists of genes, and sometimes genes that are involved in one category are also involved in another. So I was excited about that. I've not dug into these lists yet. I then looked at priming. And unlike the first Picea that you saw, where you saw a nice cluster of my triangles and my circles, you don't see anything. So that suggests initially that there isn't a difference between weevils that have been attacked twice and weevils that have been attacked once. You're still seeing some vague clustering by time point, which makes sense, but you're seeing that the circles and the triangles are evenly spread. And you see this as well when you do the actual statistical analysis. You've got no genes that are significantly differentially expressed by treatment. Plenty for time point and no for interaction. And I could have just stopped there and been sad, but instead, I continued. And I looked at it by individual time point. And I got really excited because I think I now have a story in my data. We don't have many differentially expressed genes at time point 1. But by time point 3, we do. So that suggests that these plants have a reaction at time point 1, which is the same if they're primed or not. So immediately after they've been attacked, they're all responding the same. But by a month later, they're not, which suggests potentially that these trees that have been primed are having a longer lasting reaction. So they have been primed, and now their response to this second attack is longer lasting. And I, again, took those gene lists, and you can see that some of those are in certain groups. Sorry, this is really small. But we've got response to wounding, bacteria, and a selection of other things, which was very exciting. So my takeaway points: weevils show no difference in feeding behavior. Trees do respond physiologically to insect attacks with a strong initial response which reduces over time. And there was no difference in gene expression between primed and unprimed trees at earlier time points, but there was at time point 3, suggesting priming potentially increases the duration of response. Thank you to my lab, John Mackay, and the rest of the forest tree group, Roger Moore, my secondary supervisor, all of these people. This is my funder. And a special thanks to Dave Colatello and Alvin Youngchuck, who have provided me seeds, which I'm doing my field trial with, which I didn't talk about here, but is still very exciting. So thank you.
Thank you very much, Barley. That was very interesting. I actually have one question. In the seeds provided, did you get any of the resistant? Yeah.
So this field trial that's ongoing here, nice and cold in Scotland, I've got, yes, both resistant families and susceptible families to white pine weevil, but I'm also looking at provenance material from the whole native range, just in case the resistance isn't necessarily conferred, but it could be found somewhere else.
Right. Awesome. All right, we have time for one more quick question. Is anyone? Sally?
Nice talk, Barley. Thank you.
Thank you.
When you looked at the, when you had a table that looked at up versus down-regulated genes early on, you don't need to go back to it. But when you looked at it overall 3 time points combined, you saw more genes that were down-regulated than were up-regulated. Was that a surprise?
A little bit of a surprise. And I've spoken to John about it, and it is surprising, but something being down-regulated could be something not related to response. They could be down-regulating growth. They could be down-regulating all kinds of other pathways. It was unexpected, but I think it's not necessarily as shocking as I found it initially when I was like, response should be everything turned on.
All right, we're going to switch to Richard here. Richard Sniezko is from the USDA Forest Service in Oregon. He's giving a talk on growth and survival of western white pine seed sources in the face of white pine blister rust and a changing climate after 15 years.
Okay.
Take it away.
Hi, thank you. I've enjoyed the meeting so far, looking forward to the rest of the talks and everything like that too. I'm going to talk today about a success story for forest genetics, and not basic research, but actually getting something out to the field and being successful. Going to talk about an exotic pathogen, white pine blister rust, not native to North America. It affects 9 of our species. Actually, I'm going to have material that I show from 3 different programs, British Columbia, the Interior West, and then our program in Oregon and Washington, so you can see how generally applicable it is. and working in conjunction with Washington Department of Natural Resources. It's interesting how these trials came to be, and I don't have time to go into that, but with a couple pathologists there, we got a series of trials out across the landscape in western Washington, and so that's what we'll talk about. White pine blister has an alternate host, which is ribes and some other species and stuff like that too. It doesn't really damage the ribes, but it can kill a lot of the pines. And then some of my co-authors are listed at the bottom there. So white pines and white pine blister, there's 9 species native to the US. 4 of those are also in Canada. They're all highly susceptible to white pine blister, which came from Eurasia about 100 years ago. One of the species, white bark pine, you heard some stuff earlier. It's listed as endangered in Canada, listed as threatened in the United States. And then ribes are the main alternate host. And they've tried to do a lot of things to, resistance breeding provides solutions. So that's what we're really about. I'm not in the research branch. I'm not even in the forest health branch. I'm actually in the national forest branch, and our job is to get a solution and get it back out there. So what I'm going to show you, I'm going to show you a lot of results that look really good. But what I can't tell you, and what some of you all will be able to tell, and people like Jun Liu, is come behind our material, look at material, and tell us why it works. Okay, it works. I think it would be durable resistance. I think for the most part it would be stable resistance. But we don't know the number of genes. We don't even know all the underlying mechanisms. But it works. And the good thing about resistance is it's kind of an organic and green solution. You know, we're not putting chemicals out there. We're just rebalancing the equation. I think this will work for most species. I think this could work probably for emerald ash borer and ash. I think this principle could work probably for chestnut and the program that I mentioned this morning that somebody else was involved in, et cetera. So genetic resistance, really the phase 1 you ask when you get into a program, is there genetic resistance? And you might think, okay, hey, that's easy to ask. Let's go ahead and look at that. But it's amazing how many times, in one of the programs I work with, Porterford cedar and Phytophthora lateralis, pathologists spent 20 years looking at it. It's in the literature that there is no resistance to Phytophthora lateralis and Porterford cedar. They were about to give up. They had 2 plants that didn't die. They retested them. They found resistance. And they kind of mischaracterized it a little bit, but that's kind of where the whole operational program with Porterford cedar started and stuff like that. And then that was one of the fastest moving programs, maybe the fastest moving tree resistance program in the world. And there's actually quantitative resistance and qualitative resistance in Porterford cedar. So you got to be careful when you answer that first question. You got to make sure you have a good assay, be cynical, et cetera, too. Then you want to ask, what is the frequency and geographic distribution of resistance? What type of resistance? Is it major gene resistance? Or is it quantitative resistance? Is it usable level of resistance? Is it going to give you something above 0% survival? And can you deal with it if it gives you 30% survival? Or do you need 100% survival? We're not crop breeders. I'm not in the business of finding a cultivar that gives me 100% resistance and putting out a single genotype. I want to put out populations of trees. So I want my population to be better than the high susceptible ones that don't sustain themselves on the landscape. And then you have 2 fundamental questions when you have resistance, and that's what we're going to get into here. Is it durable resistance? You know, the pathogen or insect has the capability to evolve, and in many cases in crops, you hear the boom and bust cycle where they put out something usually with major gene resistance that lasts 5, 10, 15 years. It becomes overcome by the pathogen or insect, and then they have to put out a new cultivar. So is it going to be durable resistance? In forest trees, our species have to be out there for decades, hundreds of years, and really many of the species we talk about have to be the progenitors of future generations. So really, the resistance we're looking at has to be durable. It has to last forever, okay? The other thing that most people don't talk about is stable resistance, okay? And most people aren't really familiar with that term. Is it effective over all environments? Will it be effective with climate change? Little known kind of secret that in crops, if you change the temperature even a couple of degrees, many of the resistances in some of the crops, especially like rust, they're no longer as effective as they usually are. In some cases, they're no longer effective. And so certainly when since I'm trying to solve a practical problem, I don't want to have stuff put out there and say, I got durable resistance, except it's not going to be effective under climate change. So we have a couple trials going on in our greenhouse right now to test things under temperatures, and there are some stability problems with resistance to a certain extent. Are there fitness costs? That's another question. Are you going to lose growth and adaptability, with resistance? And then how do you effectively set up a program? What is the future of western white pine? White pine blister is really serious. Climate change is coming along. And so some of the data, we put out these trials about 15 years ago, and we'll have data over a wide range of sites. This is the map on the left shows where the 6 trials are, so they're pretty well scattered over western Washington. The numbers in parentheses will show the percentage of trees of the susceptible family, the high susceptible family, that get infected by age 15 at each of the sites. And you can see for several of the sites, it's up close, 4 of the sites, it's up close to 100% or at 100% infection. So 100% of trees are close to that get cankered. The number in the parentheses is what it looked like in, I think, let's see, it was in, in 2011, so at the first assessment earlier. So some of the sites, that site, the Suwa Jigsaw site, even at age, I think 5, 97% of the susceptible trees were infected with cankers, and then eventually 100%. Whereas some of the other sites, it took Russ a while to find the site and to really come up, but many of the sites are now 90 or 100% infected, and then the low one down there, Caveman in southern Oregon, or southern Washington, is 57% infected. And this is just showing a graph showing over time the rate of infection in the susceptible check lot. And perhaps the graph below it is even more interesting, because you can see a lot of the sites went close to 100% infection, but there's one site, the one up in the left-hand corner, Suwa Jigsaw, that has many more cankers per tree. Averages 20 cankers per tree up there too. So to me, that's the site I really want to know. How does our resistance hold up under not only a site that the susceptible gets 100% infection, but the susceptible gets all sorts of cankers on it. Almost a worst case scenario. This is just a photo of some of the sites and some of the cankering and whatever too, and then the growth. We actually had an abiotic event here at one of the sites, at one of the assessment years. It looks like it hasn't continued, but it was either a cold event or a heat event. We had a couple of pathologists they never really settled on what caused it. But for the most part, the trees have recovered in subsequent years and stuff like that too. So these sites are essential not only for white pine blister resistance, but also they're sentinels for what happens under climate change. We have seed sources in this trial from central Oregon all the way to British Columbia to interior Idaho and stuff like that too. So we can ask the question, how adaptable is white pine over the landscape? Is major gene resistance durable? Okay, it was found out a long time ago, actually in the 60s. It wasn't really published until the 90s for various reasons that are almost political. But there's a major gene for resistance in white pine blister rust. However, in Western Oregon, we don't rely a whole lot on it, because that photo up there, you'll see some red trees and some green trees, okay? The red trees you might think are a susceptible control, but they're actually the major gene resistant family, okay? The green trees are the trees with quantitative or partial resistance and stuff like that too. So technically there's, I think, 3 families at that site, I think, with major gene resistance. It's essentially 100% mortality. Our susceptible control is actually a little bit better than that. And then most of the trees are living, so it's a beautiful demonstration of quantitative resistance. It's a beautiful demonstration of be careful of using major gene resistance in tree species, because it will eventually bite you. Now, I say that, but you're going to see in Washington the story is different for some reason, and we'll see how eventually it plays out. So on the graphs on the right show the percentage of trees that have stem symptoms at 6 different sites in most of the major gene resistant families in Washington, not in Oregon, but in Washington. You can see for the most part, at most of the sites, you have some families that have very low level of cankering or stem symptoms and everything like that too. And the family on the top that's shown in the axis is actually, it has a parent that is big R or homozygous dominant for the major gene. So it should have no infection at that site. So you could say, Rich, well, so does that mean you have virulence there? Because it does have some infection. And we don't really know the answer to that yet. I think what's happening up there, because in our greenhouse with some temperature trials that we have, you can erode the resistance in the major gene resistance family by changing the temperature a couple degrees. So I think what we're seeing there probably, and it's interesting, it's not an all or nothing thing, because when you put trees out there as opposed to a cultivar, every seedling has a different genetic background, even in the same family. So I think there's some genes that help condition. So I think 20 or 30% of those families, this major gene resistant family, homozygous dominant parent, kind of the resistance erodes. It's a good thing that not 100% of it erodes. So the story is still evolving. Okay, there are 4 Canadian lots in there. John King or someone sent these down a number of years ago and excluded, and I can give you the ideas of them later. 2 of them actually involve some parents from Oregon or Washington crossed with some BC trees too. But if you compare to the susceptible control, the BC lots do a lot better for percent cankering. They're, they do a lot better, but they still have a fair amount of cankering. The other thing you're going to learn here, I'm going to show a graph of survival in a minute. And so just because a tree gets a canker doesn't mean it's doomed or anything like that too. In the susceptible family, it pretty much does mean it's doomed. But in these resistant families, partial resistance doesn't, just getting a canker isn't the whole story. So here's a susceptible control, 2 major gene resistant family, our top quantitative disease resistant family, and then the Bingham orchard lot, F2 orchard lot, which is probably the most widely planted orchard lot of western white pine. And in here you can see this is percent cankering or stem symptoms at age 15. The susceptible, especially at the Suwa jigsaw site, 100% of the trees are infected. You can see the Bingham lot is up around 80% of the trees infected. Our quantitative lot, you can see, is maybe about 35% of the trees that are affected. And then the 2 major gene resistant families that are displayed there are below 10% infection. So, to a certain extent, we're great, we're going to plant major gene resistance. And at least through the age 15 in the state of Washington and Western Washington, major gene resistance is looking good. But because of our experience in Oregon, I don't know how long that will continue. In these particular trials, we're going to continue to follow them. One problem with following them now is the trees get big. A lot of infections on a lot of sites are relatively low in the tree, below about 10 or 12 feet. So one of the questions I have about the major gene resistance, even if they hold up for the next 50 years, they may be beyond the threshold for the most part of where infection sites are on the tree. They're self-pruning, so many of the branches below 10 feet are self-pruning. You won't have a lot of fights. So if these things don't get infected high up, it will look like major gene resistance lasts forever in the state of Washington. You would really have to put new trials out in and continue with that. So you have to be careful in your interpretation of that too. This is stem symptoms over time for partial resistance in quantitative resistance family versus susceptible control. And you can see the Bingham seed orchard site here in the middle. And at the dotted line is a high hazard site where there were like 20 cankers per on the susceptible family. So you can see, at that site, the Bingham gets up to about 80% infection. Our susceptible control gets 100% infection. The major gene resistant family, let's see, I don't have that here, but our best quantitative lot is over there. Even at the high hazard site, it only gets 35 or 40% infection, stuff like that too, in terms of cankering and everything too. So some of the quantitative resistant family, and we've tested this family many times, both at Durena in almost every test that we do, and also in the field as well too. So, part of the takeaway message here will be quantitative resistance works. We just need more of it and to maybe continue to build it up. Major gene resistance we can couple with quantitative resistance. By itself, I think you have to be really, really careful. Sooner or later, I think it will break down. These are just 6 different families showing the trend at different sites. We've just started really the summary of this. Ben, who's also in this room, is working on that with me. And we've just started looking at this data. So, probably 6 months from now, we'll try to write it up and we'll have it more complete. So this is an initial look at some of the things. This is just, if you take, the dotted line is a susceptible family over the 6 sites. And if you say, okay, Rich, you got these trees that are cankered, some of them are 80% cankered, 90% cankered, how bad are the cankers? We have a severity scale from 0 to 9. 0 is there's no cankering. 9 is it's dead from rust. Anything with 5 and above means that rust is girdling around the tree, and then depending how much it's elongated and stuff like that too. So you can see the susceptible family at almost every site is of the 6 sites, the dotted line is above all the other families in the trial. So every other, there's 36 families in these trials. All the families are better than susceptible at almost every site. Even in terms of severity of infection, you see a red line along the bottom. That's one of the major gene-resistant families. The severity of infection at several of the sites is 0 because a couple of the major gene-resistant families have no infection at a couple of the sites. So a lot of variation among families. The graph on the right, if we look at just the high severity site where the susceptible family got 20 cankers per tree or whatever like that too, and on the right, that shows a rust severity of 9. All the trees are dead. I think technically, actually, there's one tree still alive, so I've got to update that. There's a major gene-resistant family, and there are a big R one, which is a red kind of square, and you can see the severity of that is like under 1. There's a couple other places where there isn't even a dot because there's 0% severity because there's no infection even at this really high severity site. So the major gene resistance at that site is holding up. At that site, it appears that there's not even a stability problem of major gene resistance, whereas some of the other sites, there's still, so we want to look at some of the environmental variables across the sites and see if some of the sites are milder sites. Okay. This shows mortality from non-rust factors. So one of the things we'll eventually want to talk about is, okay, great, you solved the rust problem, kind of. Are these seed sources adaptable across sites? And so this shows the level of mortality average across all sites. You can see there's some family variation, but even in the worst case, some of these sites had a lot of competition, have a lot of water flow. There are some root diseases. Some of these are in very, very wet spots, in standing water and that type of thing too. So some of the sites will have, and at the bottom it shows the mortality per site from other causes other than rust can vary from 3% to almost 30% at the one site. The one site, the first time our technicians went in, they took machetes because the blackberries were above the head and they had to carve tunnels so they could get to the trees and stuff like that too. It's still pretty bad, but it's not quite as bad as that now. And then overall survival, this is what really comes down there, both from rust and other factors. The thing to notice there, the red bars are the major gene-resistant families. So, you could say, well, great, we're going to find major gene resistance in western Washington because they have the highest survival, and that is true. The big question is how long will that last before virulence evolves like it did in Oregon? The green line, the green bar over there is our top quantitative resistant family. So you can see it's almost up with some of the major gene resistant family in terms of total survival. And then, a lot of blue bars are just variation in the other families. And remember, this combines both rust mortality. But what you can notice is the susceptible, which is on the far right, there's no bar there because they're essentially all dead. So all 35 families in here, including some of the orchard lots, have higher survival to different degrees than the high susceptible control lot. So, there's resistance within white pine. It varies by family or orchard lot. Just because you got a canker doesn't mean it's going to die. And then for the most part, these hold up across sites. What not showing here, because we didn't have a chance to look at the data, we also have growth data, height and diameter growth data to a certain extent. And we've moved some seed lots from central Oregon up to all over Washington, and some of them are the best looking trees and the biggest trees. So I don't know if that's a climate change thing or whatever, because I think in the early provenance data, it kind of showed the northern populations were better growing and whatever too. But some of the biggest trees are from the Champion Mine area, which have the major gene resistance. So, you could look at the data and say, hey, your biggest trees are the major gene resistant families, and they're from central Oregon, so we're just going to use those. But again, because of various things, you want to be a little bit careful on that. the high hazard site is that one at the top, and you can see that the susceptible has essentially no survival, and then several of the other families. The Bingham F2 orchard lot is at the average of all the other 36 lots put together. So it's good, but it's not nowhere near the best. And so in summary, all seed lots do better and susceptible for resistance. There's no breakdown yet of major gene resistance, but you want to be careful into the future on that. There may be some non-stability of resistance in some of the MGR lots, but it's not an all or nothing. It's probably a percentage of the seed lot. There's some very high quantitative disease resistance families, and that helps us ensure that we may have durability of resistance over time. I think you can plant western white pine even on high hazard sites if you use the right material. These trials will continue to be monitored, but there may be few new infections expected because the trees are getting big, and at least on these sites may be outgrowing where rust would infect trees. That's not true on all sites. And we'll continue to monitor these trees for growth, because which seed sources up in Washington will be best under a changing climate, or will it matter? A lot of people are involved in getting these trials out there. A couple of industry groups have established a couple new orchards up recently in northern Washington. A couple new field trials by industry are being put out in Oregon, et cetera. And then just acknowledge a lot of people are involved in this type of work. This is probably, could be the top success story for a non-native pathogen in a native tree species, probably anywhere in the world, in terms of it's not perfect, you're not at total immunity, but I don't think that was ever the target anyway. All right, thank you.
All right, thanks. Thanks, Richard. That was a great talk. Does anybody have any questions for him? Oh, man.
Here, just as you pointed out, the bulk orchard lot didn't perform as well as the best full sib family, which I think would be true in almost any program for almost any trait, right? But I think what I saw on your, this is more of an operational angle on it. It looked like the Bingham orchard lot did better than the other orchard lots that are west side.
Yeah,
which is a little bit contrary to what we might be concerned about based on some papers.
It did, but you have to realize the history of things. The Bingham lot was a very highly selected lot, as you and I talked about the other day, some inbreeding in there and some advanced breeding. The two lots that are in Oregon and Washington were the initial base selection that was put out there. There's a lot more genetic diversity. So really, they're almost quasi-clone banks for breeding that are serving as orchards. And so now the next step is, hey, a lot more genetic diversity in there and whatever too. So we do have the BLM at Horning has a Horning orchard lot, which I think is going to be the top currently producing western white pine orchard lot anywhere. It's going to have more diversity than Bingham lot, et cetera, too. So it's good that you caught that, because that is certainly true. It's interesting, in our screening, when we screened the other two orchard lots, they didn't have very good survival. So I'm saying, oh, okay, well, they're better than susceptible, not very much. But actually out in the field, you know, our screening is probably maybe too harsh on these things. And because they're young seedlings, 2 years old, pretty luxuriant and stuff like that too. So you even have to be, I was actually encouraged by the field results of the two orchard lots there, because in our screening, they're not that, you know, they maybe have 5% survival. Yeah.
Okay. Question in terms of durability. Have there been or are there any ongoing trials challenging these families against different inoculum sources from, for example, Oregon versus Washington?
Yeah, we routinely, when we test our western white pine, we often use the virulent strain of rust against the major gene. So we wipe that out to begin with, and we look to see what's behind that. So we look to see if we can have quantitative resistance. And then we put these things out at a whole bunch of different field sites. Like here, there's a bunch in Oregon. There were a couple in Northern California. Nick has our material at least at 4 of his sites as well, too. So we have them over a range because we want to see how they will hold up. You got to realize that at Drina, where we're screening, we're at low elevation. We're giving these things a lot of fertilizer. We're giving them a lot of water. And I think over the years, our major gene resistance lots, I've noticed, well, they perform inconsistently. And I think we've noticed the stability over years, and we just haven't known why. So I think the virulent strain of rust for white pine for the major gene exists from about our facility, Dorina, to maybe almost the Oregon border.
Yeah, thank you, Richard. How do you assess or quantify stability?
Yeah, stability is, for instance, one would be a couple of the white pine lots have major gene resistance. They have homozygous dominant parents, so their expectation based on our nursery tests are no infection. Okay? And so if you put them on a site that doesn't have the virulent strain of rust, and they show infection at different levels, then the question comes, is that an instability of the resistance? Now, what we haven't done with this site and would like to do, and I've got to talk to Ben about it and whoever else, is these 6 sites out there, what are the climatic parameters out of those sites, and which of those sites show less stability, and is that tied to milder sites with more rainfall? So we haven't got to that point yet. Not all major gene resistant families falter to the same degree. So obviously genetic background going along with a major gene influences that as well too. And that may come into things such as co-adopted gene complexes and whatever too, things beyond which we'll look at. So in our case, it's not stability in terms of necessarily, well, I guess it is kind of G by E, but in a different respect. For the quantitative lots, it's really easy, because if we have a homozygous dominant, we're expecting 0. If something falls from 0. With the quantitative lots, then it's more nuanced, where, hey, the Bingham lot suddenly is high. The Bingham lot at one of our sites down in Oregon has 99% infection, okay? And the interior west will tell you it's only supposed to get 33% infection or something like that on many other sites. But we have a site, and then Rich Hunt up in low elevation British Columbia also had a case where essentially it failed.
All right, we're waiting for lunch a bit, so is any other questions? Did you have your hand up? All right. So that's it. Oh.
Yeah, I have one more quick question. So I apologize if I missed this, and this is my first introduction to the work you've done, so thanks a lot for the overview. You talked about finding major gene resistance in the families, but I'm curious if you've done any genomic resources in the pathogen and characterized diversity in the pathogen, and maybe interactions between at the genomic level.
Yeah, good question. We've done relatively a little bit with Junjun Liu with the Canadian Forest Service. We're starting to look at things like that a little bit. But, and the Cornartion rubicum, from what I understand, the genome's just been sequenced, so that should help as well too. But that's where we now have the materials that researchers and people with genomic tools can start looking at that more explicitly. Too often people look at material early on, don't talk to the tree breeders or whatever that really have the good material, mischaracterize things. There's numerous examples out there too. We're having people come to us and say, okay, you have well-defined resistant material. You have a couple strains of the rust or the other pathogen. They can now put it together and understand why this is working. They may even be, by understanding why it's working, they may be able to say, hey, due to the nature of things, we think it's going to be durable because it's this many genes, it's these type of genes and stuff like that. I can't tell you that. All I can tell you empirically is holding up over time. We have some trials of sugar pine that are 40 years old that people put in the mid-'80s. I'm following them. I was out one of them recently. There's some trees, major gene is holding up in southern Oregon for sugar pine. And then there's some quantitative resistant trees. Some of them there's a lot of trees that are dead, but there's some trees that have cankers this long, but they don't grow around the tree. And so, and hey, the tree's grown fine, and it's survived through 40 years, and now it's a matter of whether it will survive under climate change and stuff like that too. So there's different nuances that come into play with resistance, and too often people curtail that early on, or too often researchers are using the wrong material.
Sure you don't have any more to share with us, Richard? Okay.
I do, but it's over a glass of wine at the thing tonight.
Yeah. All right. Well, thank you, Richard. Thank you, Tia, for moderating the session as well. Thank you. Just a quick announcement, and I'll wear this. I didn't re-communicate to to the staff to communicate to the kitchen to have lunch ready at 11:40. So you got some more free time. But we will try and get back on track. So it'll be a speed pizza buffet. There is the gluten-free, vegan options as well. So again, if you didn't ask for that, just leave those for those who did. But in the meantime, use the opportunity to network and check out some of the posters next door. And then, when you smell the pizza wafting out of Ballroom 2, then you can wander in and enjoy, okay? Very good. So thanks again. We'll make an announcement at lunch to get us back into our plenary session with Brian Cullis after lunch. Thank you.